रीएजेंट्स और सप्लाईज के हवाले से हम बात कर चुके हैं जिनकी हमें ज़रूरत पेश आएगी फॉर हाइब्रिडाइजेशन ऑफ द प्रोब विद द डीएनए। अभी हमें बात करनी है कुछ स्पेशल इक्विपमेंट की विच इज़ रिक्वायर्ड फॉर हाइब्रिडाइजेशन प्रोसेस। सबसे पहले वी नीड एडहेसिव डॉट्स which are marked with radioactive ink or some phosphorescent adhesive dots. We can avoid the use of these dots, but if we use them, it will help us to align the membrane and the X-ray film for future reference. What we can do with these dots, if we suppose this is our membrane, and if we remember that we talked about making a cross on the top right or top or bottom left end of the membrane so that you can in future see the alignment. If you don't do this edge cutting, then you can use these dots to place at different positions of the membrane, and when you place the X-ray film over it, these dots will give us the position of the dot and alignment of the X-ray film. Like for example, if I place these dots only on the top left end, and that mark is going to be observed on the X-ray film as well, I will get an idea that this is my top left side, the other one is the top right, and so on. Then we need some hybridization container. The hybridization container are of three different types which are in use. The first one is the sealable plastic bags. The second one are the glass containers, and the third one are the plastic bottles. For each of this hybridization container, different conditions are used. But we are going to talk about the hybridization container for hybridization process because these are easy to handle and these are not going to contaminate the system or the working area. Then we need incubator, a special incubator which is designed for hybridization. We need incubator shaking water bath at 65 degrees centigrade if we are using phosphate SDS buffer as hybridization buffer. As you can see in this picture, this container is the hybridization container specifically designed for hybridization process, and these incubators, these are specifically designed to use for hybridization. For these incubators, these are constantly rolling within these incubators so that the probe, the hybridization liquid, constantly passes through the membrane surface and has equal access, the probe has equal access to the DNA sample which is going to be hybridized with the given probe.